So whatever the supponent was there was having the virus outer coat, and that's the reason what happened was the sulfur incorporated in the protein, and protein coat was there outside and it could not go inside the cell. Let's see the next part simultaneously. The other test tube, what they were doing in there, what they had done was they actually took radioactive virus once again, and radioactive virus with phosphorus, and we all know phosphorus is in the nucleic acid. So when this nucleic acid was labeled and then infected with bacteria, and then when this supponent and the bacterial radioactivity was checked, in case of phosphorus, it was seen, the radioactivity was seen in the cell and not in the virus. That means whatever the injected material was there was a DNA, because the phosphorus was going in a DNA, in the nucleic acid, and this after the reaction or after the experimentation, the radioactivity was seen in the cell and not in the virus. That means whatever the bacteriophage was injecting into the cell, and because of that the hereditary information was passed on to make the copies and all, was a DNA. This is how Hershey's and Chase was ultimately had got an answer, evidence that the DNA is the hereditary material, and this fact was supported with this evidence. And then actually basically previously every McCarty had already discussed this, but was not given any evidence in organisms, and every time scientific evidence has to be supported for giving any facts in the particularly in the science, isn't it? So this is what was Hershey-Chase experiment. Now when Hershey's and Chase experimented, and lastly after a year you can see that Watson and Crick gave the double helix model of DNA, which we are going to see in the next video of mine. We have actually studied this in the previous class in 11th standard in biomolecules, nucleic acid. The structure of DNA is in detail taught to you, but I'll just brush up in shorter in the upcoming video of mine. There you will see how Watson and Crick has given the double helix model in 1953, and you will get you have actually got a clear-cut idea of it, but still it will give a support-free information to you. Okay, now this is what was all about the Hershey-Chase. Now children, I'm going to as I had discussed with you all long back, if you remember, I had told you that I'll be asking you certain question when I am talking about Griffith's experiment, when I am talking about every McCarty's experiment, and lastly Hershey's and Chase experiment. So I'll be asking certain questions. You have to take down this question on paper or maybe your notebooks, because the answers, yes, of course, will be provided to you in the next coming video, but I want you to solve yourself and pass it to me in the answer to me in the maybe WhatsApp and all. Okay, so let's finally then let's take down certain questions. Okay, so define bacterial transformation. I will go a little slower. Define bacterial transformation. Who proved it experimentally? Who proved it experimentally, and how? I repeat my question. Define bacterial transformation. Who proved it experimentally, and how? Is that clear? Shall I repeat it a third time? Okay, let me repeat it third time. Define bacterial transformation. Who proved it experimentally, and how? Alright? Okay. Now, next question, children. Take down the second question.
Turning the clock.
So, the second, here comes the next question for you. I'll be also providing the answers. But till then, you have to make your own answers and send it to me. I remind you once again. Who performed the blender experiment? Who performed the blender experiment? What does this experiment prove? who performed the blender experiment? What does this experiment prove? Describe the step followed in this experiment. Describe the step followed in this experiment. Alright? If you focus my video clearly, you will get the steps very easily. So that's it for today. Take good care of yourself and see you the next time in next upcoming video of mine. Thank you very much. Take care.